To carry out the normalization of 1 and 2 color agilent data, first we will consider the 2 color agilent data. For the 2 color agilent data, the simple process for background correction is subtraction. We use the subtraction, and in this process we simply minus or subtract the background intensity with the intensity of the spots. The only drawback of this process is that when we subtract the background intensity, sometimes we get the negative value after subtracting from the spot intensities. To overcome this problem, there are many tools that when we use for the correction of background intensities, we always get the positive answer for all of the spots of the probes. One of this process is named as the norm_max_p with the offset value of 50. So this offset value is assigned a value to the intensity of the spots so that it facilitates in the process of background correction. And the second problem we face in the 2 color agilent data is that of the dye bias, because we use 2 different dyes of different colors. One is red and one other is the green, but the weight of both of these dyes are different. So when we use these dyes to as a chromogen and dye the samples, so this difference in the dye color, it hinders in the proper processing of the data. So to overcome this dye bias problem, we have another tool that is known as the lowest normalization. To use when we use the lowest normalization, the problem of dye bias can be overcome. And in this lowest normalization, it fits a curve to a data, and this curve is then used to normalize the expression of the genes. And we can use the command normalize within arrays, and then we can within brackets we can add the data. And secondly, this dye bias lowest normalization can be used in conjunction with the norm_xp, because we can use this one. Here you can see we can use the library lemma, then we can use the dat2, and because we have to use the lowest, because if in the dye bias we have to use the normalize within data array, and then we use the method that is lowest method, and we can use this lowest method in conjunction with the norm_max_p and with the offset value of 50. So the resulting normalized data is stored in an object data2 that is type MA list. So for the 1 color agilent data, the same process will be applied. That is, we have to do the background correction and then the data will be normalized. But we cannot visualize the data for the 1 color agilent. And for this process, we need to normalize the data, that is the raw data, and then after background correction, we will do the normalization, and then after normalization, we have to convert the data into the log2 transformation. So in this case, in the 1 color agilent data, we have to do the 3 separate functions one by one: background correction, normalization, and the log2 transformation. And in this case, the normalization that we will use is the quantile normalization. So we will see here the 3 steps. First is the background correction, and for background correction we use the tool that is, we have already seen, norm_max_p with the offset value of 50. In the second step, we will use the command that is normalize between arrays, and the argument we will use is method, that is quantile normalization. And thirdly, after this 2 steps, we have to transform the data into the log2 values so that we can analyze the data further.